And I'm happy now to introduce the first speaker, because we have here with us Jean-Yves Madec from the French Agency for Food, Environmental and Occupational Health Safety. And I think everybody working in antimicrobial resistance, he knows him, because he's a really very busy person, active in many areas and also in a lot of committees. He's a doctor of veterinary medicine and also a PhD. And I just will not spend very much time. Welcome to us, and the floor is yours.
Okay, so, oh yes, can you hear me? Okay, thank you, Annemarie. Yes, so hi everyone, and many thanks to the organizers for inviting me. I'm very happy to be here physically, and also for this very nice birthday, so happy birthday to BFR. I think my challenge will be to keep you awake here. But more seriously, yes, the organizers proposed to me to highlight a bit the link between AMR and the environment. It's not that obvious, because when we think about AMR, what comes to mind to everyone is the use of antibiotics, the excessive use, inappropriate use of antibiotics in human medicine and veterinary medicine. But you will see, and I hope I will not cover everything, but you will see probably that the environment has also a role in this cycle, and especially in the current moment where we are all talking about One Health, you will see that the environmental dimension of AMR is also very important. And I will start my talk with 3 little stories. Yes, just to highlight a bit for you how the environment is present in all these kind of stories. This is a story of NDM1. NDM1 is very popular and is familiar to everyone working on EMR. It's a new daily metallobanctamase, very important in human medicine, causing troubles, treatment failures, blah, 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 at a world scale. But you can see that NDM1 genes were also found in the environment, in tap waters, in surface waters, and also in very remote places in Norway, in the soil of Norwegian archipelago. So here you can see here that despite the fact that NDM1 is a very big issue in human medicine, there is a kind of environmental component here. A second story is this very funny tropical fish and this vast contamination of human beings all over the world by the same strain of Salmonella, multidrug-resistant Salmonella paratifi B. Coming from a tropical fish, all the fish were contaminated, they were coming from the same batch from Singapore, and then expand the dissemination of this isolate. We didn't have whole genome sequencing at that time, but with those PFG gels, the authors could easily demonstrate that the same strain was coming from one place and disseminate everywhere. So the tropical fish is an animal, of course, but it's very close to the environment. It's really the environment. So these EMR genes are present, and these bacteria are also present in the environment. So there is also a very strong environmental component here in this story. And the third story I like very much is this MAC-C MRSA story. You may know that the resistance to methicillin is driven by one single gene since the 60s, the MAC-A gene, and suddenly in 2011, scientists discovered that a new gene called now MAC-C gene was also responsible for methicillin resistance. And this gene was discovered in cattle and humans in UK and Denmark, so it was quite a very nice paper, scientific paper, but as you can see in The Guardian the day after, the message was the new strain of MRSA superbug may have spread from cattle to humans, and the people in the UK and everywhere were thinking that they could get MRSA by drinking milk. So this was a mistake, by the way. But the story is not finished. Actually, some colleagues, and especially from northern country, and I can mention here a good colleague and friend of mine, Stefan Bergersson from Sweden, they have observed that the prevalence of MAC-C, MRSA, was quite high in hedgehogs. So hedgehog has nothing to do with dairy cattle and humans, but many groups have discovered that, and the story was resolved this year in a very nice paper, and I encourage you to read the paper. Actually, this is a combination of a story between three friends: the hedgehog, a parasite called Trichophyton, and the Staph aureus, which is a bacteria. And the Trichophyton produced penicillin, so if for the Staph aureus he wants to live together with the two other colleagues, he needs to be resistant to penicillin, and the mechanism was MIC for him during the evolution. So it's a nice, very nice ecological story. And you can see here, that's nothing to do with the use of antibiotics in dairy cattle and the transmission of MRSA to dairy cattle to humans. And here you can, I think, see how important is the environmental component of AMR. So after these three stories, just to summarize, yes, of course, we know that AMR is One Health, and of course we need an integrated approach to tackle this issue. But the first point, as long as we're talking about the environment, what would be a definition for the environment? You can see it here in this picture, the interplay between the different sectors, and if you look at the environmental sector, it includes many different things like rivers, streams, drinking water, aquaculture, soils, whatever, wildlife. So it's very diverse, and this is one of the difficulties in working on AMR in the environment. And one important aspect of the environment is precisely this dual aspect. It means that the environment is both a kind of source of reservoir of AMR genes and antibiotics. You know that antibiotics were found in the environment at the beginning, very beginning. And it also, the environment can be polluted by AMR genes, AMR bacteria, and antibiotics. So this dual aspect is very important to understand if you want to place correctly the environment within the AMR topic. So the first point, the source of, we all know that antibiotics and AMR were present in the environment well before the use of antibiotics by humans. And of course, this questions a lot of things, like the role of antibiotics in nature, to which concentrations the bacteria are exposed to antibiotics in nature, and so on, and how all these antibiotics, bacteria, and genes evolves jointly. This is a nice example of antibiotics coming from the environment. Here you have a nice frog producing an antimicrobial peptide. Also, you know that many environmental bacteria produce antibiotics. This is a nice paper, proof of concept that you could get antibiotics from environmental bacteria. So this is a reservoir, a source of genes, and those genes may, to a certain extent, be a kind of danger for us, because we have proof and evidence that some of those genes have jumped from the environment to clinics, to common bacteria like E. coli, like Salmonella and others, that cause problems in clinics. And this is the case, very famous case for those people working on AMR, of the CTXM genes, which are present in environmental bacteria, and one day they have jumped, we don't know when, why, and in which conditions, have jumped to E. coli, and now have expanded very broadly in humans, in animals, and also in the environmental sector. So this is the first point. The environment is kind of a source. And the second point, vice versa, is the environment can be polluted by AMR and by antibiotics. So I will not go into a lot of details on the pollution by AMR genes, but there are tons of papers showing that you can find AMR genes and AMR bacteria in the environment that were selected in humans or in animals. I've taken the example of ESBL E. coli, which is a kind of very important multi-drug resistant bug. And you can find those ESBL E. coli. ESBL stands for extended spectrum beta-lactamase, producing E. coli, in wildlife, in soils, in wastewater, in seafood, and so on. So this is the kind of pollution, right? This is a work we have conducted a couple of years ago, where we have found very important multi-drug resistant bacteria in seafood, and those seafood were close to the wastewater of 4 hospitals in Tunisia. So the bugs were coming from the hospital and directly to the seafood. Not nice? Yes, this is a contamination. A recent work we have conducted were on fecal deposits from dogs in Lyon. I live in Lyon and I love the city. I invite you in Lyon if you want to visit Lyon, and you will find fecal deposits from dogs everywhere in city parks. And we have demonstrated that and provided evidence of transfer of bacteria, contamination with plasmids, AMR plasmids, in those fecal between dogs, between different unrelated dogs through the environment. And another interesting story also I would like to present today is this work we have carried out 2 years ago from a rescue center for white birds. Okay, the white bird entered the rescue center without any AMR, but they are released in nature afterwards with a 25% increase of AMR. I mean, 25% of the animals were AMR carriers. So this also questions the impact on the environment when you release animals that were more or less treated within a kind of hospital and get out in nature being carriers of AMR in nature. So all this is pollution with AMR genes and AMR bacteria. And at a more global level, of course, you know that almost 500 million people defecate outdoors. This is clearly an important information, especially in certain countries or continents. And wastewater is a major issue for AMR, especially in those conditions. And here I can refer to a study, a very nice study from the group of René Hendriksen in DTU in Denmark, where they conducted metagenomic analysis in wastewater, and they could evidence different patterns of resistance depending on the region and continents and the places, the locations where they looked at the content of AMR in the wastewater. Okay, so pollution is okay with AMR genes, with AMR bacteria, but also with antibiotics. So I can encourage you to read this article from this year, published this year in PNAS. It's not only on antibiotics, but it's very interesting for you to know about the levels of pollution by pharmaceuticals in general. And you can see at the very beginning, the 5 or 6 ranked, first ranked molecules are trimethoprim and sulfamethoxazole. And those two are antibiotics. So it means that there is also a pollution of the environment with antibiotics. And this is particularly true in certain regions, like in Southeast Asia, where sulfonamides are dominant in sewage in Southeast Asia. Also, oxytetracyclines are dominant there in livestock and aquaculture waste. And then pollution is coming further from the rivers to the sea. So there is clearly a pollution by antibiotics. This is also nice work, and all the works from this group are very interesting and important. The group from Joachim Larsson in Sweden, showing that downstream of discharge sites, you have very elevated concentration of antibiotics you can detect in the environment, and especially in those countries like here in India. And this is a work we have conducted in France at ANSES, where the question was on the environmental contamination in France by antibiotics, AMR genes, bacteria. And there are certain conclusions that you can read here on which indicators to choose if you want to estimate the contamination of the environment by AMR and antibiotics, and also on the methods and strategies. But as long as we are talking about pollution, there are 2 questions, at least 2 questions, maybe, probably more, but that comes to mind as well. One of them is what levels of antibiotics are needed to select AMR in the environment. And you know that in the environment, bacteria are not free living. Most of them are in biofilms. So it's not completely obvious to imagine or to know, to try to decipher which levels of antibiotics are needed to select AMR. And there are this kind of work on this issue, showing that, for instance, certain concentrations are needed to select for resistant to ciprofloxacin in E. coli in those complex aquatic biofilms. Also here you have a nice study on trying to decipher the concentration of tetracycline which is necessary to select AMR in complex community here in freshwater biofilms. So these kind of questions are very important when you are studying AMR in the environment. And also another interesting question from a scientific point of view is, okay, we release antibiotics, but we also release other pharmaceuticals. And the question is, do the other compounds, other molecules than antibiotics select for AMR, co-select for AMR? And I will show you a couple of examples. I will not go into detail for each of them, but here, for instance, you can see that heavy metals like copper or zinc also co-select for AMR. It's not only antibiotics that select for AMR. And those co-selecting factors are also present in the environment. So this is a nice study on AMR from marine biofilms on painted and unpainted surfaces. But you have the same kind of global results with other molecules like triclosan, which was shown to have an effect on the transfer on sulfonamide-resistant genes in a complex sewage community effluent. Also the same from petrol and diesel. It's quite funny. Can exhaust the conjugative transfer rate of AMR. Also the same for fluoxetine. Fluoxetine has nothing to do with antibiotics. It's an antidepressant, but it induces resistance to multiple antibiotics in E. coli. This is also a nice paper, recently published. Also polycyclic aromatic hydrocarbons. They also accelerate the propagation of AMR. Metformin. Metformin has nothing to do with antibiotics. It's, to my best knowledge, is treatment for diabetes, I think. And it promotes AMR development in E. coli. So, as you can see, the environment is a very diverse and complex sector. And those kind of issues can be highlighted. It means that other chemicals that can also select for AMR, even though they are not antibiotics. So I would like to finish up my talk by kind of words on risk assessment. What about risk assessment related to AMR in the environment? Do we have arguments for human exposure? And I think this is one of the major parts for which we don't have many scientific data. This is a paper highlighting the fact that probably when you surfers are more at risk to get ESBLs compared to non-surfing people. This is one of the single papers looking for human exposures, but I can refer to you those two papers were really trying to decipher the attributable sources for certain resistant bugs coming from different sectors, including the environment. But the data from the environment are so limited that it's difficult to integrate this in the more global approach. Maybe an idea. This paper has nothing to do with antibiotics because it's about carbamazepine. But I think we may try to mirror this kind of approach with antibiotics to try to really get more data on the human exposure from whatever contaminated with antibiotics, but taking example from other type of sectors like contamination here with carbamazepine. A question you may have heard about is whether manure application is at risk to spread AMR. And I took this example because, well, science is not so stabilized. And this is also an issue when we are talking about AMR in the environment. Here you have a nice paper, a solid paper from a solid group looking for tetracycline-resistant genes abundant in soil. And they report that the abundance of tetracycline increases over the years after exposure to tetracyclines. So it seems to say, okay, yes, it is at risk to spread AMR. But this is another paper from also a solid group the group from EdTop in Canada, a very famous scientist in the field. It's not on tetracyclines, it's on macrolides. It's also long-term, 8 years, and several AMR genes were enriched, but at the 10 and not the 0.1 milligram per kilo of soil. So it means that the exposure at realistic concentrations did not increase AMR genes, really. So it's not that clear. And when you consider that one paper is on tetracycline, this one is on macrolides, 2 different antibiotic families, you have different type of source, different type of context. So we still need data, really, scientific data, to inform the risk assessment and therefore to inform policies or possible policies. Another issue is whether you will get minimal environmental targets from the industry. In 2016, there were a high-level meeting on AMR, and this is a kind of commitment from the companies, industry companies, that they will go forward towards science-based targets for antibiotics in receiving waters. This is from the industry. This is a nice first step. We will see what happens, for instance, for the 2016. But yes, it's also an important point to consider when we are talking about AMR in the environment. And from the public or risk assessment domain, the question can be the following. You know that in Europe, an environmental risk assessment for a medicine is required if the predicted environmental concentration exceeds 10 nanogram per liter. And this leads to the calculation of a predictable non-effect concentration. And because it's a very general process for many types of molecules, there is a concern that this assessment is biased for antibiotics. So there is also something to improve there, because the models, the way to estimate this PNSC is not really appropriate to antibiotics. This is also something to improve in order to go further into the risk assessment in terms related to AMR in the environment. Of course, we need modeling approaches to estimate the risk of AMR development in the environment. These are 2 very nice papers from a very famous group as well, trying to estimate the risk of antibiotics present in the environment. And what is nice, it will be the end of my presentation, is that recently the United Nations have set up a global agenda to take into account the environmental dimension of AMR. And this UNEP is discussing with FAO, WHO, and OIE, or WHO animal, for OIE, so come in my mind. And UNEP is really important, is kind of, a very important milestone in the way that at the global level we will take into account the environmental dimension of AMR into the story. Okay, so my final remarks would be, and I would refer to this paper, of course, if you want to go further into this paper. We have seen that the environment is very complex, different sectors, and what we don't know really is the relative contribution of each sector. So if we want to take actions, we need to know which areas are more important or are bigger than others, to put our efforts on those sectors where the contribution is the most important. And we don't really know which are the relative contributions of the different sectors. Also, what we don't know is the role of the environment itself in the selection of AMR. Because we have seen that there is a lot of pollution, for instance, but what is the role of the environment in selecting AMR by himself? The third one is probably one of the most difficult question. What is the impact on human health? How can we measure and quantify and assess the risk for human health coming from what happens in the environment? This is also a very important issue, and we don't know much about it. And then, what can we do? to mitigate AMR in the environment. And those are technological, sociological, that's very important, economical, behavioral interventions, all kind of interventions or methods or strategies that we need to improve to mitigate the spread of AMR within and via the environment. Okay, so thank you very much. This is a cartoon. We were in the previous session on comics and cartoon and so on. So this is a cartoon in India that was drawn by a guy during conference. I was probably talking about India, and he was producing this kind of cartoon. So thank you very much.